listeners, welcome to episode 133 of Cell and Gene, the podcast. I'm your host, Erin Harris. We know that precision oncology has transformed cancer treatment over the past decade. That's a fact. But many patients with advanced solid tumors still have few effective treatment options. And increasingly, researchers are looking beyond targeting cancer mutations themselves and instead are exploiting the biological vulnerabilities that those mutations create. And so in this episode, I'm joined by Dr. Oren Gilad. He is the Chief Executive Officer of Aprea Therapeutics. He and I discuss how the company is advancing a pipeline of precision oncology therapies targeting the DNA damage response pathway. You'll hear how the science behind synthetic lethality and why biomarker-driven patient selection is essential to the future of targeted therapy, and how Aprea is developing a next-generation WEE1 inhibitor designed to improve both efficacy and tolerability. He and I also look ahead at where precision medicine is going and what it will take to bring more effective therapies to patients with difficult-to-treat cancers. This is a great episode. I'm really excited for you to check it out. Definitely share it with your peers and colleagues. It's well worth your time and theirs. And so without further ado, please check out my conversation with Aprea Therapeutics, Dr. Oren Gilad. Enjoy. Hi, Oren. Welcome to Cell and Gene, the podcast. Thank you so much for your time. I'm thrilled you're with us and looking forward to telling Cell and Gene, the podcast audience, all about Aprea Therapeutics and its mission. So why don't we go ahead and start with that? Tell us everything that Aprea Therapeutics is working on, its mission, and your role.
Thank you, Erin, and thank you for inviting us to share Aprea with your audience. Very excited about that opportunity. And at Aprea, we can't wait to cure cancer. And we do it by targeting multiple targets in the same pathway, namely the DNA damage response pathway. We have a WEE1 inhibitor, we have an ATR inhibitor, we have a p53 reactivator. Those are 3 clinical programs. And we have one preclinical program, the DIRC1. And together, our mission is to cure as many cancer patients as possible.
Terrific mission. It doesn't get much better than that. And so let's dive into how we're going to, will it make that possible? So, okay, precision oncology has obviously transformed cancer treatment over the past decade. Still many patients whose tumors don't respond to currently available targeted therapies, however. So how do you and your team see the next generation of precision medicine evolving? And what would you say are the scientific advantages of really making that possible?
So I think if you look at the way the oncology field is evolving and shaping, targeted therapy, precision medicine is very exciting time right now. Because I'll give you one example, and I think it's an important example to really show how next generation precision medicine is evolving and the scientific evidence that making it possible to target that, as you mentioned, patient that consider untreatable, right? So one example is a RAS mutant in metastatic pancreatic ductal adenocarcinoma, PDAC in short. There's a company called Revolution Medicine, and they have a very great drug for that indication and that mutation. If you go back only a few weeks ago, another company called Tango Therapeutics, they combined their PRMT5 inhibitor for MTAP deletion in pancreatic cancer, and this combination received 92% overall response rate. That's a very strong response. So what is allowing that is really accumulation of scientific data and smart people who are leading the way.
Terrific. Okay, so speaking of, targeting mutations, let's talk a little bit about that. So your strategy focuses on identifying vulnerabilities created by specific cancer mutations rather than targeting the mutations themselves. So talk us through that. Explain that concept and why targeting these vulnerabilities may offer advantages over more traditional targeted therapies.
So that concept is called synthetic lethality. And synthetic lethality is exactly what you described, because there are mutations in the cancer cell that are not present in the normal cell. And that is what we hope provide a wide therapeutic window. Because when you have mutation in the cancer cell, there is now another pathway that is now being active to compensate for the loss of function of this existing mutation. That's how cancer survives. So it doesn't die, it just compensates for it. By identifying that pathway and targeting it, that's, we hope, will provide a wider therapeutic window. That's the approach that we're taking with all of our molecules. So for the WEE1, for example, we have cyclin E overexpression, platinum-resistant ovarian cancer. We have uterus serous carcinoma, independent of the mutation, because there's just such a high replication stress in these patients that we don't need to select. We have PPP2R1A as the mutation. When those are existing in the cancer cell, that means that WEE1 is hyperactive, and by inhibiting WEE1, even systemically, normal cell can live with lower level of WEE1 activation, but cancer cell cannot. And that's what hopefully will give us the wide therapeutic window.
Terrific. Okay. Let's drill down a little deeper on WEE1. So it has emerged as an exciting target in oncology, but earlier WEE1 inhibitors have encountered challenges related to safety and tolerability. So what have researchers learned from those earlier programs, and how has APR 1051 of yours designed to address those limitations?
So, Aaron, this is a great question because you touched on the liabilities that are associated with the other WEE1. And what we did before we even started the program, we went and looked at every molecule, every program, every patient that has been treated with it, preclinical data as well. And we have identified another group of enzymes that is not WEE1 related. It's a PLK. It's another group of enzymes that seems to not have selected for. So the other WEE1 seems to inhibit both WEE1 and the PLK family. So we went back to the drawing board and we created a molecule that is very selective to WEE1 with minimal effect on this family of enzymes, the PLK. And that's what we saw, that's what we're seeing. Hopefully we'll continue to see the differentiation, because it seems to also show potential increased tolerability and increased efficacy. We saw the first PR in a patient at the 100 mg level. So we did see stable diseases at 70, but then, was it 150? And then we saw it at 220, a confirmed PR, when the target lesion shrank by 50%. So what we think we did is that there's sometimes advantage of not being the first, and we took advantage of that. We saw, because WEE1 has been shown to be a good target. WEE1 has been shown to shrink tumors. But the challenge was exactly as you described it, was tolerability. It wasn't on the efficacy, it was on the tolerability. So how did we solve it? We solve it biochemically by identifying the liabilities, addressing it, and hopefully the trend will continue.
Okay, that's great. Now you're evaluating APR 1051 in patients with advanced solid tumors that harbor specific genetic alterations. Mm-hmm. How important is biomarker selection in maximizing the likelihood of clinical success? And I guess second to that, what have you learned so far about identifying the patients who may benefit most from this approach?
The biomarker selection is the key for success or failure of targeted therapy, including the WEE1 and every other targeted therapy that is out there. Because the field is shifting from chemotherapy that is killing every dividing cell and causing horrible side effects into targeted therapy that is biomarker-driven. WEE1 is a good target for patients with this specific genetic mutation, like overexpressing cyclin E, and mutation PPP2R1A, FBXW7, KRAS P53. It's not likely to work on any kind of tumor with any kind of mutation. So being able to understand the science, being able to really enroll the patient that are most likely to benefit from this therapy is key for the success. And that's the approach. PPP2R1A, the two patients that I mentioned previously that had the partial response, one had the tumor shrinkage 50%, the other one had 50% in the first scan, another 10% in the second scan. They both had a PPP2R1A mutation, and they're both endometrial cancer patients. So the study is still ongoing, and the ability to enroll the right patient population is key for the success, because then what the ultimate goal is to have the efficacy with very minimal side effects and very minimal toxicity. That's what determines the therapeutic window.
Sure. Okay. And speaking of, minimizing toxicity, we know that one of the biggest challenges in oncology drug development is achieving that meaningful tumor control while minimizing, of course, toxicity. And from your perspective, I guess both from a scientific and clinical development side, how do you even strike that balance, and what characteristics would define a truly best-in-class WEE1 inhibitor?
I'll start with the second part of the question. The truly best-in-class WEE1 inhibitor is going to be defined by few parameters. One, single-agent activity. You want to show that your molecule works on its own. It's also important to know what kind of toxicity to look for. In the case of WEE1, it's likely neutropenia. That seems to be the on-target effect, but the key is going to be preventing neutropenia into progressing into sepsis and death. Combination therapy, because combination therapy is important in oncology. Lots of combination are taking places. So having a clean molecule that can work in combination with other standard of care and do not have overlapping toxicities, that's also going to be important. Because when you do have two molecules with overlapping toxicity, whether hematological toxicities or other toxicities, it's just not helping. It's going to make it worse. It's not going to make it better. And we've seen that, I don't know how many times, over and over and over again. So having a molecule that has a clean safety and efficacy without overlapping tox with other standard of care, that's another factor that's going to determine truly best in class. And of course, at the end of the day, shrink the tumor, kill the cancer, and don't have much effect on it, don't have much side effects.
Terrific. Okay. Before I let you go, talk to us a little bit about where you see in the next handful of years, what's on the roadmap for Aprea, and then from a field-wide perspective, how do you see precision medicine, whether it be from a clinician or researcher perspective, or perhaps even, just your perspective as your role at Aprea? Where do you see it going over the next few years?
So Aprea is in an exciting junction right now, exciting times, having the portfolio that we have. That is really, and we have a slogan that we used in our corporate deck, one critical pathway, multiple targets, right? So if we go for the WEE1, we're going to continue with the dose escalation, continue enroll patients, We're probably going to start combination at one point, and hopefully everything would translate nicely and we cure more cancer patients. For the ATR, we already said that we're looking for a combination, and we're looking for partners to combine our ATR that also has very clean safety profile and attractive mechanism of action for combination therapy. There are two holy grails targets in oncology. One of them is RAS, and we talked about Revolution Medicine. They have a good drug for RAS. The other one is p53. And Aprea has APR-246, which is a p53 reactivator. This molecule had a very successful phase 2 and went to phase 3 and came short. So we already made a public announcement that we're going to be looking at the patients in the phase 3 and hopefully identify a patient that did respond, did benefit from the treatment of this drug. So that's on the 246, and the preclinical program, the DIRC1, it's still preclinical. We're working on in vivo proof of concept before we progress it into IND and hopefully into the clinic. So that's what's coming. And I think from the overall oncology drug development, ADC are becoming, very important approach for cancer therapy. Aprea also has this next generation ATR inhibitor, which is very important, that can be used as a dual payload on ADC, which we are exploring. So I think combination therapy, ADC is in the RAS, of course, that we talked about, Revolution Medicine. So we're probably going to see more combinations with this drug in different indications. GI is going to be probably another one of them. And so that's where Aprea also plays within the whole oncology strategy for therapeutics.
Excellent. Excellent. Well, that about wraps up this episode featuring Dr. Oren Gilad, CEO of Aprea Therapeutics. Oren, thank you so much for your time. This has been a tremendously insightful episode, and our Cell and Gene podcast listeners are in for a real treat listening to this. So thank you for your time.
Thank you.
And thanks, listeners, for tuning in. Check us out at cellandgene.com. And please encourage your peers and colleagues to subscribe to Cell and Gene, the podcast, so they too never miss an important episode just like this one. And we'll talk to you soon.